All right, so like I said, we're going to act out protein synthesis, and the reason I like to do this is I remember a few years ago, a former student emailed me from college, and she said, I was thinking about when we acted out protein synthesis in genetics class. Because she said in my biology class at Purdue, we are right now learning about protein synthesis, and a lot of my friends are confused about it. They're acting like it's the first time they've ever seen it. And so they were having trouble with it. But I understood it because I just thought back on how we acted it out in class. And I thought, well, that's cool. Maybe it was a good idea after all to do that. And she said, in fact, in the evening in the dormitory, in the dorm, we all got together and we acted it out, just like we did in class. So that's something you guys could do for fun this weekend. Probably not. Probably not. All right. We left off on page 9. We're ready for number 6, I think, right? Number 6. Okay. mRNA. Anybody know what that is? Who said it? Good. All right. I'm going to write that down. Messenger RNA. Messenger RNA. A close cousin of DNA. Different kind of molecule from DNA, of course. And when we act it out, messenger RNA is really not going to be an actor. It's going to be a prop. Okay? So here it is right here. This is going to represent our messenger RNA molecule. And before we even get to this, let me just tell you, we're going to pretend like this room is a cell. Okay? And my office back there will be the nucleus. And I'm going to turn the light off in there. This is the nucleus. Very mysterious in that nucleus there. Nobody really knows completely what goes on in there, right? Usually there's just a lot of paper grading going on in there during prep period. Okay, that's going to be the nucleus. Now, theoretically, who's supposed to be the in charge adult in here?
You.
Me. Now sometimes people may not think that, because sometimes I act a little juvenile. And the main reason for that is to make sure people don't fall asleep, okay? Actually, there probably is a lot of truth to that. A lot of times when I'm in the presence of adults, they kind of don't know how to act. And I think, why are these people acting like old people? Because I'm not used to that. I hang out with young people, right? So I don't act my age sometimes. All right, so who's supposed to be in charge in here? Me. And so I'm going to be the DNA, okay? I'll play the part of DNA. And I happen to have a DNA costume right here.
That's pretty weathered.
Pretty weathered. Tried and tested. This is my DNA costume. So you see, Isaac, if I walked up and down the streets of Lafayette wearing this,
You'd be the coolest guy on the streets.
that's right. I would have other stuff on too. Yeah. And I would be walking down the street. Okay. That's pretty obvious, right? Okay. If I'm walking down the street like this, everybody looking at me would know that I'm DNA, right? Noah?
Yes.
I think you're a Boy Scout.
Or could be a scout. Or Miss
Or
America. No, probably not.
Or DNA.
Mr. Jeff. Or DNA. Cole, look at me closely. Can you see the letters on here?
Yeah.
Maybe not in the dark where you are.
I can see.
Okay. How can you tell I'm DNA by looking at me and not RNA?
Because of the the letters, right? There's pairs.
The what? I think you said it right. Yeah, they're in pairs. I'm double-stranded, right? Isaac, were you getting ready to say something? Oh, you're just stretching?
Yeah.
I'm double-stranded. That's one clue. What's another clue? Oh, I have thymine. See the T's? I don't have U's. RNA has U's, right? And DNA has T's. So that's a giveaway, right? People would look at me, anybody on the street would look at me and know that I'm DNA. Okay, but for right now, I want to take this off, and we'll put it right here. That's going to come in handy later. Now, during this little drama that we're going to do, I'll be stuck in the nucleus, because that's where DNA is, right? You all are out here in the cytoplasm. Now, back to messenger RNA. You might say, for a definition to write down, the molecule made by DNA. Go ahead and write that down. The molecule made by DNA. Are you writing that down? The molecule made by DNA that carries the DNA's instructions to the ribosomes. Okay. Now, if you take a sneak peek at page 10, vocabulary word number 9, there it is, ribosome. See? So, messenger RNA is a molecule made by DNA, carries the DNA's instructions to the ribosomes. And we'll define ribosome here in a little bit. Yeah, okay. Messenger RNA. So when we get that thing started, and I'll be DNA in the nucleus, I will make this messenger RNA molecule. It's going to come out here, and some ribosome is going to read it. Some ribosome. You guys remember ribosomes from biology? Do you remember that?
You learned about it
You learned about it where and when?
in anatomy.
Oh, in anatomy. Well, that's good. Little cross-fertilization between courses there.
Yeah, everything's linking up.
Everything's linking up. All right. My wife and I, for those of you on the show, we're going tonight. Is the first night usually the best night?
It's usually the worst.
Usually the worst.
I think it's the best.
Sunday nights. No, it's
I would
Saturday.
personally say Saturday works would be,
Sunday?
It just starts at about maybe a little bit of nervousness, but it's not going to be. It's not like it's going to be completely void of love.
So Sunday you really have to reach back, right?
That's the day we will say everyone's really tired.
Okay. Let's flash back to biology.
Oh no.
Parts of the cell.
Oh no.
Okay. So messenger RNA is going to carry the DNA's instructions to these little cell organelles called ribosomes. Remember all these organelles?
Yep.
Everybody seems to remember the mitochondrion.
Oh, yeah.
It's the powerhouse of the cell. Come on.
Powerhouse of the cell, right?
Yeah.
See these tiny little dots here?
Those are ribosomes.
Those are the ribosomes. Yeah, they're tiny, thousands of them in a cell, but they're probably, well, it's hard to say what's the most important because they're all important. But even though they're tiny, they're very important because those ribosomes are the workers in this factory that build proteins. Okay? What does the Subaru plant build? Cars. Cells build proteins. They're miniature factories that manufacture proteins. And these little ribosomes are the workers in here. They're the ones that are actually stringing together amino acids to build proteins. Make sense? Raise your hand if you already knew that. If you didn't, that's okay. That's okay. I wonder if there's somebody in here Who would be a good one? I think Mario would be a good ribosome.
He's a hard worker.
Yeah, now let's come on up here. We're going to do a little sneak preview. Okay, you're going to be a ribosome.
Yeah.
And we're going to build protein. Here's your hard hat. See, it says ribosome on it. And you'll also have to wear this lab coat. Okay, now, this is a sneak preview, so yeah, you can put those back to your seat. Yeah, that's what you're going to be wearing. Those are the prop. No, they're not props. They're costumes, right? So he's going to be the ribosome. You're one of those thousands of them there in the cell, okay? And what you'll do is you'll just be positioned out here. This part of the cell out here is the cytoplasm. So you'll be right here, and you'll be taking amino acid molecules, represented by little index cards, and you'll tape those amino acids in a chain up here to build a protein molecule. That's what you'll be doing. Following the instructions written in the messenger RNA. And this came from where? The nucleus, made by DNA. Okay. We're just kind of setting the stage here. All right. Next word, codon. Maybe we should call this movie Codon the Barbarian. Has a nice ring to it. No, bad movie. Okay. I want to show you what a codon is. Anybody know what a codon is?
Heard it.
You know what a codon is?
Doesn't it like tell it where to start?
Yeah, there is a codon that tells you where to start. Let's write this down. A codon is a group of 3 bases in the messenger RNA. You can just say mRNA. A group of 3 bases in mRNA that codes for an amino acid. Time out. How do we know this is RNA right here?
Single-stranded.
Single-stranded, and it contains U's instead of T's, right? This is messenger RNA. Every group of 3 bases on here is called a codon. So, Emily, that first codon there is AUG. See that? Can you guys see the AUG? That's the first group of 3 bases on this messenger RNA. So what's going to happen is the ribosome, Mario the ribosome, is going to read these bases 3 at a time. Like, for example, you're going to go AUG. And that's going to basically tell you what amino acid is going to be first, because it's the first codon here. Okay? Y'all with me?
Yeah.
Why don't you take a sneak peek at page 16? Let's take a sneak peek at that. Okay, there are the 20 amino acids I showed you last time. And these are all the different messenger RNA codons that code for those amino acids. Look familiar?
You're,
You did some of this in biology, didn't you?
yes, I think we did this exact chemistry.
I think you're right. You mean the acting it out. But I don't think Mario was the,
I think in our class.
He wasn't the
class I was in, it
ribosome.
was George.
George was the ribosome?
Yeah, George enzyme.
George enzyme, yeah, that's right. All right. So for example, that first codon I read, what amino acid does it code for?
Methionine.
Good, methionine.
Yes.
Here's AUG. Oh, methionine, right there. So Mario, that's going to tell you methionine will be the first amino acid molecule in the chain of amino acids that you're going to string together, which will become the protein. Okay, let's continue setting the stage. What's the next one? Oh, rRNA. Pirates would have fun saying that one. rRNA. Anybody know what that stands for? Good. Ribosomal RNA. Ribosomal RNA. You want a definition for that?
Yeah.
Here's a non-textbookish definition. That's the stuff ribosomes are made of. That's the stuff ribosomes are made of. Got that? You got that? Okay, it's the stuff ribosomes are made of. Ribosomal RNA. So Mario, basically that means that as the ribosome in this drama, you are a wad of ribosomal RNA. And some protein. Okay? All right, moving on down, page 10. Page 10, ribosome. Oh, what should we write for that? Mario. Or?
Manufacturer of.
mRNA?
Yeah, ribosomes build proteins. Ribosomes build proteins. They string together amino acids to build proteins. Here's another one. tRNA. What's that stand for? Transfer. Okay. Yeah. Transfer RNA. And those molecules, you ready? Deliver the amino acids to the ribosome. Transfer RNA molecules deliver the amino acids to the ribosome. They're like little taxis. T for taxi, T for transfer RNA. So Mario, you're not going to have to go wandering around looking for amino acids. The transfer RNA molecules are going to bring them to you. Hee-haw. Okay. Is this coming together?
Yes.
Noah, you look like a good transfer RNA molecule. So, let's see, here we go. Okay. You all look at Noah. Noah, come on up here. Stand up here and face your accusers. Or your classmates. All right, not Mario. Noah is a transfer RNA molecule, and Isaac, he's going to carry this amino acid right here. Methionine. Got that? This is what you're going to deliver. When it's your cue, when it's your turn, you float through the cytoplasm, irresistibly drawn to the ribosome. That's just kind of how it goes. And then you're going to deliver this amino acid to him. Okay? Now, transfer RNA molecules like this fine specimen of a transfer RNA right here, they're made of A's and U's and C's and G's, right? But there are 3 of them that are sticking out like in a strategic location, kind of like a name tag like this. And so you need to kind of clip that onto your shirt somewhere, wherever you can. Maybe on your hood string or on your collar right here. Yeah. I've seen people put them on their glasses, you know. Okay. So, yeah, that's like your ID or your name tag, because on a real transfer RNA molecule, there are 3 bases. In this example, UAC. They're sticking out. They have a very functional role. They're in a prominent position, and they're very important. Now, see, what's going to happen is when Mario the ribosome, you stay right here. When Mario the ribosome reads the codons here on the messenger RNA, like the first codon is AUG, that's going to be your cue, because your name tag says UAC. It's the opposite of AUG. See that? This U is going to be attracted to this A. That A is going to be attracted to this U. that C is going to be attracted to that G. You got it? Make sense? And it's that attraction between your name tag and this codon that will pull you up here to the ribosome. Base pairing is important. Make sense? Now, don't go back yet. Since this name tag that he's wearing, these 3 bases that are important on this transfer RNA, since they're the opposite of this codon, what would be a good thing to call them? Right, anticodon. Right there. That's the next word. Anticodon. Okay, you can go sit down. The anticodon is, this is what you might want to write down. 3 bases on the tRNA that are attracted to a codon on the mRNA. Okay, 3 bases on the tRNA that are attracted to a codon on the mRNA. And that's an anticodon. Anybody else wake up this morning thinking, gosh, I really want to be a transfer RNA molecule? Okay. All right. Lauren, here you go. Lauren's anticodon is GCC. Wear it proudly. GCC. And the amino acid that you carry is this one. It's called arginine.
Hey, Dad, I can pronounce.
Arginine. That's up close to the top of the list, isn't it, on page 16? Arginine. There you go. So when it's your turn, you're going to deliver that amino acid to the ribosome. And let's see. Anybody else want? Who else wants to be a tRNA? I don't want to pass anybody up to
T.
Oh.
T.
Chloe. There's your anticodon. Wear it proudly. And you're going to carry this amino acid, glycine. And I think I'd better get Isaac. He looks like he'd like to be a tRNA. Here you go, Isaac. Itzhak. Isaac, there's your anticodon. And you wear it proudly. And then you're going to carry this amino acid, valine. CAU. That is not Cornell Alumni University. That is cytosine, adenine, uracil. Okay. Who else did you say should be a?
Taya.
No.
T.
Taya, yeah, okay. Taya, here's your anticodon. Clip it on. You're going to carry the amino acid cysteine. And
T.
Caleb,
Oh.
there's your anticodon. That's what you have to wear. And you will carry the amino acid alanine. By golly, that's the first one on the list, isn't it? And let's see. Tierra. There's your anticodon. You're going to carry the amino acid threonine. Jonathan, here's your anticodon.
I'm wearing it very
And you get to carry the amino acid histidine. And, all right, I need one more.
T.
Diana,
I wasn't really
okay.
paying attention.
Diana, here's your anticodon, GGU, and you're going to carry the amino acid proline. Got that?
I'm out here a lot.
Okay. All right. Now then, before we act this out, you know how in most businesses, factories or businesses, they might have a photo on the wall of the CEO or the founder of the company or the big boss or whatever. So I think we should do that in here. We're going to put the boss's picture right up there. Let's see if we can find it. There it is. Okay, that's me. That's me, right? DNA, I'm in charge. I'm the boss. Everybody can look at that picture and Moving on. Okay, so, Mr. Ribosome, are you ready?
I am.
All right, come on up here. And just to keep us on the straight and narrow, I think perhaps we should put page 16 up here. Make sure we don't make any mistakes. So you guys who are transfer RNAs, you want to listen for the codon that is the opposite of your anticodon, right? Because that attraction between the codon and your anticodon is what's going to pull you up here to the ribosome. Actually, we shouldn't say opposite. In the language of genetics, we say complementary. Your anticodon is complementary to the codon on the messenger RNA. So, all right, we'll turn the lights on so nobody gets sleepy. Because I know that you guys who are in the Christmas show this weekend are probably wiped.
Yeah.
Aren't you? Wiped out.
One of the girls was
Sleep deprived.
one of the girls.
Good job. All right, so Mario, come up here. What you're going to do is, when they bring you the amino acids one by one, just take this tape and stick them up here and make a chain. And it can bend. Okay? And, all right, we're going to start from the top.
Picture college. Mario, picture college.
He's all right. He's a working man. Okay? We're going to start from the top.
Is there a messenger RNA?
This is messenger RNA right here. So messenger RNA is not an actor, it's a prop. Okay?
Someone carry it.
Oh, you'll see how this works. This is high-tech
Animation.
animation. All right. I need to suit up also. I'm DNA. Okay, I'll get back in the nucleus because that's where I belong. And we'll start from the top. Now, I'll tell you what. We should dramatize this, right? This is a drama. So here we go. Ready? Okay.
Do we need to get the camera on you?
Alas and woe, I'm a DNA molecule stuck in the nucleus. How's that?
That's pretty good.
Keep my day job. I want to make protein. Bad. But, okay, here's the problem. Cole, here's the problem. I have a lot of book knowledge. I mean, look at my base pairs. I've got all the instructions needed to make proteins, but I'm not very handy. He's handy. Okay, he can build proteins like you wouldn't believe, but he doesn't have any instructions. So if I'm stuck in here with all the information, and there's this gulf between us, I'm in the nucleus, he's out here in the cytoplasm, suggest a solution to this problem that I've got.
Send a message.
Send a message. Now I could text him. Eli, is that what you were going to say?
Yeah.
I could text him. Mario, I could text you, but instead I'm going to do it the old-fashioned way. I'm going to write you a letter. Okay? It's called messenger RNA. Okay, just hang on a second. This takes a while. I'm making messenger RNA.
TMI.
I'm done.
Oh.
Wait, you know what that reminded me of? I've got little grandchildren. They know how to go to the bathroom by themselves, but not completely. They don't know how to finish the job very well. So when they're done, they go, "I'm done!" You know what that means? Yeah, you have to go help them. Okay. All right. Watch this. Quinn, guess what?
What?
I can't leave the nucleus, but messenger RNA can. Here it comes. It's floating out into the cytoplasm. Now, pretend like you don't see me. You're going to green screen me out or whatever. Here's mRNA floating through the cytoplasm, messenger RNA, and it attaches to the ribosome. And the ribosome's going to take it from here. Okay? So I'll just fade out Okay, ribosome, take it away.
AUG.
The first transfer RNA has delivered his amino acid. We're off to a great start.
Oh, yes. Okay. GCG. Give them both? Oh, no, just one. You want both?
All right, we've already assembled 2 amino acids.
GUA.
Guac.
GUA. Here comes the third transfer RNA molecule. Needs a little more ATP, I think.
Yeah, definitely.
Wait, what did you read? What did Mario read?
GUA.
GUA?
Go.
Oops, that was the wrong transfer RNA.
We just made cancer.
We just made a mutation.
Oh,
What should be attracted to GUA? C what? CAU. CAU.
No, he said GUA. I was just yelling.
All right, now we're rolling. All right, time out. Time out. Mario, you need to know this. Okay, this is a little motivation for you. All right? A real ribosome can string together about 40 amino acids per second. So we've got, there's an improvement we could make, right?
Yeah. Okay. CCA.
I'm not suggesting we go that fast, though. Here comes transfer RNA molecule number 4.
CAU.
Well done. Now you're supposed to float back out in the cytoplasm. See?
There you go.
There we go.
I had to do a double take. You already got it.
It happens.
UGU. ACG. GGC. CGG.
What did you read, Mario?
CGG.
C, G, G.
I'm GCC.
Yeah. So that's.
Didn't read myself.
Wait, what's the? What?
No, I'm GCC.
Yeah, so you, that's,
Oh,
okay.
You're the opposite, so that would attract you up there, see?
I am confusion.
UAA. UAA, is that it?
Well, go ahead and stick that one on. Okay, so how many amino acids long is that so far? 9? Okay, what was that last codon, Mario?
UAA.
UAA. So what would be attracted to UAA? Okay, UAA.
Stop.
Actually, if I look
The
at
very
that
first
code,
one.
can you guys find a UAA on that code chart on page 16?
Stop.
Right, there is no UAA, so that means stop. Yeah, that's called a termination codon. It does not code for an amino acid, so Mario, when you come to something like that, you just stop. So you did it. You built the protein. It means UAA is a termination codon. It means end of message. Stop. End. Complete. So the protein you built is right here. It's 9 amino acids long. Well done. Good job. You can disrobe. Take off your costume. Oh, somebody left an anti-codon up
I did, I
here.
was still leaving together.
Oh.
It was a good step away.
I was going to say, go ahead, you guys, self-amputate your anticodons and pass them up to the front
I did it.
because I can reuse those. Your anticodons. In this day of budget constraints, I need to recycle the paper clips. You know? Thank you. Thanks. All right. Thank you kindly. Thank you. Thank you. All right, you guys. So that's how it works. Let's dim those lights again. A little bit. Do what? Keep the lights on. I'm going to turn them back on here in a little bit when you go to the computer. Okay? But just a little bit of a review of what we've done. What we've done. All right. Our protein is 9 amino acids long. What's wrong with that picture? 9 amino acids long. That's pretty short. Lots of proteins. A lot of small proteins are hundreds of amino acids long. Some are thousands. But you get the idea, right? So, when you're studying for the test at the end of the unit, if you're trying to remember what does DNA do, who do you think of?
You.
When you're trying to think, what does a ribosome do? What do you think of? Who do you think of? Mario. If you're trying to remember what is messenger RNA, what do you think of? This. You guys have to engage also. This, right? This is mRNA. If you're trying to remember what do transfer RNA molecules do, who do you think of? Yeah, all you guys who delivered amino acids to the ribosome. Cole, that should make it easy to remember that stuff. Right? Should make it a piece of cake, so to speak. All right. 2 more terms I want us to look at, then we're going to go to the computers. So let's go back to page 10. Let's see. Page 10. 2 more, that's 9. Where did I put page 10? 10, where's the 10? X marks the spot. Page 10. I have misplaced page 10. Rats.
You had up there.
There it is. All right. Page 10. 2 more terms. Transcription and translation. Who wants to give us a short, sweet, non-textbookish definition for transcription?
DNA going into RNA so that RNA can exit out of the nucleus.
Good. Good. Or you might say DNA making messenger RNA. Yeah. See, right here kind of summarizes what we just acted out. And it's so important, I think I've put it up here twice. Okay. This little summary right here, this right here, DNA makes RNA, RNA makes proteins. That is the central, main idea that all of biology is based on. It's called the central dogma. Central dogma of biology. If you ask anybody in the world, well, ask some people in the world, what's the central dogma of biology? And they're going to tell you, Diana, they're going to tell you, DNA makes RNA and RNA makes proteins. That's the central dogma. Reminds me of a song. Who let the dogmas out? Woo, woo. No, that's dogs. Central dogma. What's a dogma? Is that a vocab word anybody's ever had in English or anything?
Probably.
Maybe looking.
What?
Yeah, maybe that's the religious purpose.
It can be a religious idea,
Like the main idea.
but it doesn't have to be. Yeah, it could be a main idea in any field. Yeah.
A phrase or a belief that you hold to be true or govern.
Good. A phrase that, or belief that you hold to be true, that everybody in your circle pretty much agrees is absolutely true, right? And some people may be so convinced that it's true that we say they're dogmatic about it. But that's the central dogma. Central dogma. DNA makes RNA, RNA makes proteins. Now look carefully at it, because you're going to have to memorize that. Eli said transcription is when DNA makes RNA. See that? So go ahead and jot that down. And Quinn, what would you say translation is?
It's when RNA makes proteins.
Right, when RNA makes proteins. That's called translation. So if you're making copies of some kind of a document, let's say you have some kind of a document that's in English and you make a copy of it in English, another copy, that's called a transcript. And the process is called transcription. Okay? Oh, that reminds me. Yeah. Several years ago when I went to Israel, we went to the Dead Sea. We went among these caves near the Dead Sea. It's a desert environment where people like 2,000 years ago, they were called Essenes, and they stored scrolls in clay jars in these caves. Some shepherd boy then discovered them. I think it was 1947. I better check that. And they contained all these scrolls that were 2,000 years old. They were called the Dead Sea Scrolls. So there was a community of people who lived out there, and they were very very religious type people, and they left Jerusalem because they thought the leadership in Jerusalem was corrupt. And some of the people in that sort of colony out there near the Dead Sea their job was to copy the scriptures, make copies of the scriptures. Listen to the STEM word script, okay? They would make copies of the scriptures. And we even saw ruins of the little rooms that they would do that in. And those little rooms were called scriptoriums. And the people who did that, who made the copies by hand, because they didn't have photocopiers, they were called scribes. So the scribes would copy the scriptures, make copies. That was called transcription. Because if you have a copy of writings that are in Hebrew, and you make another copy in Hebrew, you're in the same language, right? So that process is called transcription. See why we call that transcription up there? If DNA is making RNA, you're kind of staying in the same language. The language of nucleic acids. DNA and RNA, they're both in the same class of molecules called nucleic acids. But what's it called if you take writings and you make copies in a totally different language? Translation, which is what it's called when RNA makes proteins, because RNA is in the language of nucleic acid molecules. Proteins are in the language of protein molecules. Different classes of molecules, almost like different languages. Makes sense if they would call it translation? That's how it works. So translation is when RNA makes protein. Raise your hand in this drama that we acted out if you were ribosomal RNA. Point to him. Mario. The ribosome is a wad of ribosomal RNA. Raise your hand if you were a transfer RNA. We had 9 of them. Okay, and this messenger RNA can't raise his hand, it's a prop. But these three RNAs, you see, work together to build the protein. Where was DNA during this process of translation? Just chilling in the nucleus, right? You see why I said that RNAs are probably more important than DNA? DNA is the archival copy of the information hidden away safely in the nucleus, but the RNAs are the ones doing the work. Sometimes you guys are going to hear, a lot of times nowadays, people don't call this messenger RNA. You know what they call this? A transcript. Sometimes this is called a transcript. Okay, so here's the plan. Take a look at page 11. I'm going to have you guys go to the computers, and when you get there, you want to pull up a tutorial called Protein Synthesis. And work with a partner. You don't need the headphones because I didn't put any sounds in this, okay? When you get to the end of the tutorial, you're going to encounter a video, but you don't have to watch it. Don't watch it, okay? But what's going to happen is you're going to work through this self-paced interactive animation where you kind of make the animation go. And as you go through that, fill in pages 11 through 15 in the study guide. Okay? So let me show you how you're going to get the ball rolling here. You'll see an image that looks kind of like that, and it says, describe the events shown in this diagram. So just to get the ball rolling, for this one I might just write something like, here's DNA in the nucleus. And then the second part says, in what part of the cell does this occur? Well, nucleus, right? So you're going to work through filling out pages 11 through 15 as you work through that computer tutorial. On some of the diagrams, there are arrows that you'll be labeling. Like on, for example, page 13. See the arrows? You also want to label those as you go. Okay?